In 1803, the brilliant scientist John Dalton was able to prove in the lab what the ancient Greeks thousands of years earlier thought might be true, that matter was made of incredibly tiny building blocks called atoms. Dalton thought that atoms were solid spheres like billiard balls, and that they were the smallest particles in the universe. But about 100 years later, scientists discovered that atoms were made of even smaller particles. This video shows how they were able to do that. In 1897, a clever Englishman called J.J. Thomson designed a glass tube filled with low-pressure gas and containing two metal plates inside. When he put a big electric charge on the plates, he found that the positively charged plate, called the anode, was able to pull a beam of tiny particles out of the negatively charged plate, called the cathode. Because the beam came from the cathode, he called them cathode rays, and he called the apparatus a cathode ray tube. You can see the beam as a blue line in the diagram. Where the cathode rays arrived at the anode, there was a little hole that allowed them to travel through and continue and pass between a pair of parallel plates that he could put varying charges on. Sometimes the top one was positive with the bottom one negative, and sometimes the other way round. The beam always bent away from whichever plate was negative and towards whichever plate was positive. This meant that the particles must have carried a negative charge, because we know that like charges repel and unlike charges attract. But here's the thing. Because the beams bent so easily, more than other particles going through the plates, he could work out that they must have been very light, thousands of times lighter than a hydrogen atom. Thomson realized that atoms had even smaller particles inside them, and that they carried a negative charge. He had discovered the electron. He won a Nobel Prize for this. J.J. Thomson was not only a brilliant scientist, he was a really good bloke as well, and he helped many of his students become famous scientists too. 7 of them went on to win the Nobel Prize, including his own son. One of his students was Ernest Rutherford, an extroverted New Zealander with a booming voice and a big laugh. Rutherford was playing around with beams of positively charged particles, which he called alpha particles, that were emitted from uranium atoms. He knew they were positively charged because they bent in the opposite direction to the electrons of a cathode ray when passed through charged parallel plates. They bent more slowly than electrons, so he knew they were heavier than electrons too. Rutherford used these alpha particles as atomic bullets and fired them at a very thin sheet of gold that was only about 1,000 atoms thick. On the opposite side to the uranium source, he placed a scintillation screen that lit up when particles hit it, so he could then measure where the alpha particles landed. If the gold atoms were like billiard balls, then the alpha particles would be blocked and none would arrive at the screen. Instead, almost all the alpha particles passed straight through the gold foil, as though it wasn't there. Rutherford realized that the gold atoms were mostly empty space. J.J. Thomson thought that the mass of atoms was pretty evenly smeared out within the substance, and he also thought that most of the alpha particles would pass through. But something very surprising happened. Some of the particles were bent a few degrees off course, and about 1 alpha particle in 20,000 bounced right back in the direction from where it came. Rutherford said at the time that It was almost as incredible as if you fired a 15-inch shell at a piece of tissue paper and it came back and hit you. What this meant was that the mass of the gold was not evenly spread out at all, but concentrated in very small lumps that were positively charged. He had discovered the atom's nucleus. Because the nucleus was so small, most alpha particles didn't even come close and completely missed them. They went straight through. If some went close to the nucleus, its positive charge would cause repulsion and bend them off course. If an alpha particle made an occasional direct hit, it would bounce right back from where it came, because the nucleus was much, much heavier than the alpha particle itself. The nucleus is incredibly heavy. Over 99% of the atom's weight is concentrated there, and incredibly small. If we magnified an atom to be the size of a football stadium, the nucleus would be the size of a pea. Rutherford later discovered a particle inside the nucleus that was responsible for its positive charge, and he called it the proton. An atom's overall nuclear charge was equal to the number of protons it had in its nucleus. And this also turned out to be the same as the atom's atomic number. Each element had its own number of protons in its nucleus. How's that for a coincidence? But there was puzzling evidence that the nucleus was heavier than could be accounted for by protons alone. Scientists could measure the weight of protons, and they knew how many protons were in the nucleus of each kind of atom. But when they measured the overall mass of these nuclei, they were usually about double the mass of all the protons put together. Was there another particle in the nucleus? In 1932, James Chadwick, a student of Rutherford's, discovered a beam of new particles with about the same mass as protons. He did this by shooting alpha particles into beryllium atoms and knocking these particles out of the beryllium nuclei. The beryllium target was much thicker than Rutherford's gold foil, so there were many more nuclei for the alpha particles to make direct hits with. Beryllium's nuclei has an atomic number of 4, so he knew that they had 4 protons each, but he was experimenting to find a new particle in the nucleus. When these particles were struck by the alpha particle bullets, they were knocked out of the nucleus and then continued in a beam that Chadwick could study. This beam did not bend when passed through charge parallel plates, like the electrons of Thomson's experiment did. Remember, it was the electron's negative charge that made them bend when going through the plates. This meant that the newly discovered particles had no charge. Because these particles were electrically neutral, he named them neutrons. Chadwick had an eventful life. During World War I, he was a prisoner of war in Europe, and he even did a lot of his experiments while he was a prisoner. Then during World War II, he helped invent the atomic bomb in America. By now, scientists had a fuller picture of how atoms were made. They had a nucleus consisting of positively charged protons and neutral neutrons at their centre, and this was responsible for most of their mass. Light, negatively charged electrons whizzed around the outside of the nuclei, in fact at about 3/4 of the speed of light, and gave atoms most of their size. The question of how atoms could stick together was also answered. Having an opposite charge to the nucleus, electrons are attracted to it, so the electric forces can hold electrons travelling at near the speed of light and bend them into an orbit around the nucleus. Electric forces act as a kind of atomic glue holding atoms together. They are quadrillions of times stronger than gravity. For those of you who have learnt about the powers of 10 way of writing numbers, electric forces are 10 to the power of 33 times more powerful than gravity. That's 1 with 33 zeros. In the next video, called Atomic Structure, we will look at how electrons are wrapped around the nucleus in shells, and how this dictates the properties of atoms. For example, whether they will be a gas, liquid or solid, or their colour or brittleness, or whether they will be a metal or a non-metal. Pretty much everything. In this video, we'll look at smaller and smaller objects in a series of steps. We'll start with this oak tree, which is 10 meters tall. Can you see the boy standing underneath? We'll call him Lee, and Lee's height is about 1/10 of the tree's. That makes Lee 1 meter tall. We'll now zoom in using a magnifying power of 10, and that will make Lee's image about the same height as the tree was on the screen before. In this video, we'll continue to find smaller and smaller objects and zoom into them in a series of steps until finally we can see an atom. Now let's find something about 1/10 of Lee's size. The distance between his eyebrows is about right. Let's zoom in to magnify this by 10 so it fills up the screen. Now what's 1/10 of this distance? The width of Lee's iris. That's the coloured part of his eye. His iris is 1/100th of a metre wide. 1/100th of a metre is also called a centimetre. We're going to show you a trick here. 1 on 100 is the same as 1 on 10 squared, because 100 is 10 squared. Scientists often write this as 10 to the negative 2. That's like 10 squared, except that the 2 has a negative in front of it. This negative is telling us that the squaring is on the bottom of the fraction instead of the top. The cool thing is that it's quicker to write it this way, and it gets better. 1 on 1,000 is the same as 1 on 10 cubed, so it can be shortened to 10 to the minus 3. Notice that the 3 corresponds to 3 zeros on the bottom of the fraction. What's 1 over a million? A million's got 6 zeros, so that's 10 to the minus 6. When there are lots more zeros, it gets way quicker to write it this way. We'll show both ways of writing it from now on. Now what's 1/10 of the width of his iris? Hey, did you see the flea that jumped onto Lee's face? Lee's got a flea. Coincidentally, a flea is 1/10 of the width of his iris. That makes it about 1,000th of a metre, or 10 to the negative 3 of a metre. 1,000th of a metre is also called a millimetre. And now the flea is running for cover to Lee's eyebrows. Good move, flea. Let's zoom in. Now we'll flip the angle to show Lee's hair sticking upwards. Guess what's 1/10 of the flea's length? Yep, one of Lee's hairs is 1/10,000th of a metre wide. 4 zeros on the bottom, 10 to the minus 4. This is about the smallest size that the naked human eye can see, and from now on we would need a microscope. If we pan the camera downwards, we can look below Lee's skin and see inside his body. Can you see his blood vessels? If we look inside a blood vessel, we can see that the flowing blood contains red blood cells, and they're about 1/10 of the width of Lee's hair, or 1/100,000th of a metre wide, 10 to the minus 5. They're too small to see without a microscope. Ouch! Here comes an anthrax bacterium, and it's deadly for humans. Luckily, it's rare, and in fact most bacteria are not only not harmful, they're good for you. This one is about 1/10 of the size of a red blood cell, making it 1/1,000,000th of a metre in size, 10 to the negative 6. This length also has its own name, a micrometre. Smaller again is a virus. Most viruses are too small to be seen with a normal microscope, so an electron microscope is needed. A flu virus is 1/10,000,000th of a metre in size. That's 7 zeros on the bottom, 10 to the negative 7. The wavelength of green light is about 5 times the width of this virus. Zooming in another step, we can see that the virus wall, called the envelope, is about 1/10 of the virus width itself, coming in at a staggeringly small 100,000,000th of a metre. That's 10 to the negative 8. The envelope is made out of fats that the virus gets from its host. Yuck! Inside the virus is a strand of DNA, which is its information centre. DNA is a very long but very thin molecule made of billions of atoms joined together. This is the first time we've been able to spot individual atoms. And here comes a sugar molecule, which is a bundle of 45 atoms, about 1/10 of the width of the virus envelope. 1/1000,000th of a metre, 10 to the minus 9 of a metre. Holy dooly! 1/1000th of a metre is also called a nanometre. Heard of nanotechnology? That's about making things that are about a nanometre big. Even tiny robots that can travel around your bloodstream and fix up your body. At this scale, you can see that the whole world is made of atoms. Atoms to the left of you, atoms to the right of you, above and below. This shows you what a grain of sugar might look like, containing trillions of sugar molecules next to each other. But what's between the molecules? More atoms? Air? Well, it can't be air, because air itself is made of atoms. In between molecules is nothing. Zilch. Zip. Just empty space. Scientists call empty space a vacuum. Going back to our nanometre-sized sugar molecule, we can see that the white atoms are about 1/10 of its size. They're hydrogens, and they're about 1/10 of a nanometre, or 10/10,000,000th of a metre across. 10 to the minus 10. That's crazy small. Now we know how big an atom is. What about the other elements? How big are their atoms? A helium atom is about the same as a hydrogen atom, and all the others are bigger. The biggest is cesium, at a whopping 5/10,000,000,000ths of a metre across. By the way, atoms aren't really coloured like we've shown them here. They're too small even to have colour, because atoms are much smaller even than the wavelength of light. We just gave them different colours so we could tell them apart. So is that it? Are atoms the smallest things in the universe? Not by a long shot. Atoms are made of even smaller particles, called protons, electrons, and neutrons. If we zoom up to a helium atom and look inside it, we find its electrons whizzing around the outside, and its protons and neutrons right in the centre of the atom, called its nucleus. Protons and neutrons are about the same weight as each other, and they're nearly 2,000 times heavier than an electron. That means that most of the mass of an atom is concentrated in its nucleus. We've made the protons and neutrons bigger than they really are here, just so that we can easily see them. But they're really much smaller compared to the overall size of the atom. If you imagine that an atom is as big as a football stadium, then the nucleus would be the size of a pea. In fact, you couldn't even see the protons and neutrons from this distance. We would have to zoom in with a magnification of 10, 5 times, adding another 5 zeros to the bottom of the fraction. That's with 15 zeros on the bottom line. 10 to the negative 15 of a metre. Protons and neutrons are a quadrillionth of a metre wide, also called a femtometre. What's between the electrons and the nucleus? Nothing. A vacuum. That means that an atom is mostly empty space. Since we're made of atoms, so are we. That's enough to do your head in. Anything smaller than a proton or a neutron? You bet. Quarks are a quintillionth of a metre, with 18 zeros on the bottom, also called an attometre. 10 to the negative 18. And neutrinos are a septillionth of a metre, with 24 zeros on the bottom, also called a yoctometre. That's 10 to the negative 24. Did you know that trillions and trillions of neutrinos are passing through your body right now, every second? But don't worry, they're so tiny and light that most miss your atoms completely. That's about as small as it gets. I don't know about you, but I can't imagine stuff that small. We've come a long way since seeing Lee under the tree. Then again, we now know that the whole world is made of incredibly tiny particles, called atoms. 1 million atoms will fit across the width of a hair. The word atom comes from the Greek word meaning uncuttable or indivisible. We can easily cut a strand of hair again and again and again. But eventually, if our scissors are small and sharp enough, we would cut down to a single atom inside the hair, and we could not cut that. This means that an atom is the basic building block of the hair, and that's true for all stuff in the universe too. Although scientists prefer to call stuff matter. So then, an atom is the basic building block of matter. Huge mountains are made of atoms. People. Things people make, like cameras. Even air is made of atoms. Now, a fair question to ask is, are all atoms the same? Well, the answer turns out to be no. Scientists have discovered 92 different kinds of atoms in our world. In fact, when they look into outer space, they find the same 92 kinds of atoms in the whole universe. Now that's incredible. "98% of the universe is made of hydrogen and helium left over from the Big Bang, as the birth of the universe is called. Back on Earth, there's lots of oxygen, silicon, aluminium and iron, and smaller amounts of other types of atoms too. But it's rare to find a substance made of just one kind of atom all on its own. Normally, different types of atoms are mixed in with each other somehow, like oxygen and nitrogen in air. But when we do, we call that substance an element. A substance made of only one kind of atom is called an element. Air is not an element, but a nugget of gold is. It's made of only gold atoms. So what's this element gold like? Checking it out, we find that it is yellow, shiny, soft, solid and dense, which means heavy for its size. If we had the same space filled with water, gold would be 20 times heavier. If we hammered it, we could flatten it out into such a thin sheet that a piece of gold the size of an M&M would cover your classroom floor. This is called being malleable, and gold is the most malleable element of all. Another test would show that it conducts electricity really well, and if you heated it, it would melt at 1064 degrees Celsius. These things about gold that we can observe or measure are called its properties. They are unique to gold. Let's look at an element made of carbon atoms instead. Because carbon and gold atoms are different, we expect carbon and gold elements to be different too. You don't have to go far to find a piece of pure carbon. If you have a lead pencil on your desk right now, you really do have about 100,000 billion carbon atoms in the lead. The lead isn't lead at all, but carbon. It should be called a carbon pencil. Up close we can see that carbon is black, or blackish, shiny, hard, solid, and it's got medium density. That is, it only weighs about twice the same space full of water compared to that of gold's 20 times. Hitting it with a hammer will smash it into bits, which means it's brittle, the opposite of malleable. Rubbing it on your finger will show it's slippery, or leaves thin layers of itself behind on your skin, pencil marks. It conducts electricity, but not as well as gold or other metals. If you heat it, it vaporizes at 3642 degrees Celsius. You can see that carbon's properties are different to gold's. They are both shiny and solid, but overall they are very different. It's as though each has its own personality, its own unique set of properties. And this is linked to the uniqueness of the tiny atoms that make it up. We can't see carbon's billions of atoms with our own eyes. They're much too small. But we can with the strange-sounding electron tunneling microscope. By zooming in 1 billion times, we can eventually see a single carbon atom. You can see they're a bit fuzzy. All atoms are. Now let's back up, and we can see the whole bunch of carbon atoms, which together comprises the element carbon. We can use the word carbon to mean either a single atom of carbon or the element carbon made up of many carbon atoms. Both uses are correct. If we mean a single atom of carbon, we are speaking microscopically, that is, on the very small scale. If we mean the element carbon, that is, zillions of atoms of carbon, so many that we can see them, then we are speaking macroscopically, that is, on the large scale of the everyday world. We've seen that the properties of an element depend a lot on which kind of atom it's made from. So how are these kinds of atoms different to each other? Let's line up a few and see. Some types are small and light, like hydrogen, the lightest, and others are big and heavy, like uranium, the heaviest, and other types are in between. This just shows a handful of the 92 kinds of atoms that there are, starting from the first, hydrogen, through to the 92nd, uranium. Sixth in line is carbon, and chlorine is 17th. Gold is the 79th element, so you can see it's towards the heavy end of the list. An element's place in this list corresponds to its atomic number. For example, the first and lightest atom, hydrogen, has an atomic number of 1. Carbon's is 6, and uranium's is 92, and so on. The atomic number is also connected to the number of positively charged particles called protons, which live inside the atom. But we will come back to that in a later video. Each kind of atom has its own atomic number, and each atomic number belongs to just one kind of atom. Scientists sometimes shorten the name of a type of atom to just 1 or 2 letters. Hydrogen is shortened to H and carbon to C. Chlorine can't be C because C is already taken by carbon, so it's written Cl with a little l. Now you may think that the shorthand for gold would be G or GO, but you would be wrong. Instead, it is Au. That comes from the Latin aureum and refers to the word aura, meaning the glow of sunrise. No prizes for guessing the shorthand for uranium. It's just U. This shorthand way of writing is called the symbol of an element. Here are the names, symbols, and atomic numbers of the first 30 elements. I bet you've heard of some of them before. Check them out. We'll wrap up the video by going over the main points. We saw that the incredibly tiny atom is the basic building block of matter, and that an element is a substance made of only one kind of atom, and that a property is something about a substance that we can measure or observe, like colour, and that microscopic means the very small scale of atoms that are too tiny to see with our eyes, and that macroscopic means the normal scale of our everyday world that we can see with our eyes. Do you like my drawing? Don't give up my day job.
A journey to the nano-world. The nano-world is too small to be seen directly by the human eye. Microscopes are therefore used as a window into the nano-world. More advanced microscopes than conventional optical microscopes are needed to reach deep into the nanoscale. The operating principle of the kind of advanced microscope shown here is very simple. The tip of a sharp needle is used to feel the nanostructures like a blind person using a stick. In this movie, an electron microscope is used to film a scanning tunneling microscope directly while it is in operation. The sharp tip scans over the surface and an image of the surface morphology is built up line by line as in TV. Now that we have seen the way the microscope works, we will start our journey to the nanoworld. This movie captures the growth of semiconductor nanocrystals. From such movies, scientists learn many details about the processes taking place during the growth of semiconductor nanostructures. This knowledge will facilitate the fabrication of future nanoelectronic devices. However, you can just lean back and enjoy a flight through the nano canyons of germanium on silicon. The ultimate challenge in the nano world is to zoom onto single atoms. Enjoy the flight between single silicon atoms. If the scanning tip is so sharp that there is only one atom at its end, even single atoms can be observed. See how nicely the atoms are arranged at this crystalline surface. The fantastic scanning tunneling microscope is not only used for imaging nanoscale atomic structures, but also to build predefined nanostructures atom by atom. The microscope tip is used to push the atoms to the desired sites. In this way, the smallest man-made structures are created, just for fun like our logo, or in order to build dedicated structures for research purposes. For instance, to confine electrons in these nanostructures. All the movies shown here are made from original microscopy data.
Have you ever seen an atom? Seeing as everything's made of them, you have. But have you ever seen one on its own? Over time, microscopes have become more and more powerful, allowing us to see deeper into the world of the ultra-small. Traditional light microscopes can be used to see things like these onion cells and the structures within them as they divide, pulling apart their chromosomes. But scientists have come up with a whole host of clever methods to observe far smaller things. Using beams of electrons instead of light, we can generate detailed images of chromosomes themselves. Recently, groups of scientists around the world are becoming able to see materials at the most fundamental scale, the atomic. One group from the University of California in Los Angeles have been getting up close and personal with nanoparticles of platinum, just a few nanometers across. Each of the tiny dots you can see here are actually individual platinum atoms. But researchers didn't stop at a two-dimensional picture. By imaging over 100 slices of the nanoparticle at different angles, then removing the noise with a special filter, they were able to map the location of almost every atom. The information was used to create a three-dimensional reconstruction of the whole particle in unprecedented detail. It may look blurry, but this particle is estimated to contain over 27,000 atoms, and so, like flies in a swarm, they appear to merge together. Every so often, though, we see the platinum's atomic structure align, granting us a moment of clarity. This technique is being used to analyze tiny irregularities in the structure of the particle called dislocations. Dislocations are subtle, like the misalignment of the green and red layers of atoms in this particle. But nonetheless, they can significantly change the properties of materials, with effects ranging from a change in the efficiency of LEDs to the strength of metal alloys. Three-dimensional atomic-scale imaging like this is bettering our understanding of the structure of materials on this truly fundamental scale.
What does a single cell look like? We know it's small, really small, but exactly how small is it? Let's take a closer look. First of all, what is a cell? A cell is a microscopic building block that makes up all living things. Each cell contains even smaller parts called organelles that help keep the cell alive. So, in a sense, a cell is and can be its own living thing. There are many different types of cells. Bacteria cells, for example, are types of cells that make you sick. White blood cells are a type of immune cell. Finally, red blood cells carry oxygen you breathe into parts of the body to use. Now, let's talk about how small cells actually are. The human eye can barely see even the largest cells. Usually we have to use light microscopes to magnify cells before we can easily study them. The size of a cell ranges from 1 to 100 micrometers. But how small is that? In one droplet of blood, there are over 5 million red blood cells. That's the same as the number of visible stars in the night sky, in the mountains, times 1,000. In the human body, there are over 100 trillion cells. That would be the same as filling a 30-foot wide cube with 100 trillion grains of sand. It's hard to wrap your mind around the size of a cell. Sometimes we get so wrapped up in the things we do every day that we forget how amazing it is that a system as complex as our body can have so many parts that all work together perfectly. The cell is definitely a tiny thing.
Cells are the tiny components of life that we're composed of, along with everything else alive on Earth, and maybe some things in the universe. Maybe. However, you can't see a cell, it's far too small. But how small? Let's take a look at how small a cell is. Inside the human body, it's estimated that there are over 100 trillion cells located in the body. If I had $1 for every cell in my body, I'd be 150 times richer than Bill Gates. However, let's try isolating a random cell and looking at it. There it is. Oh, can't see it? That's right, cells are invisible to the human eye. In order to properly see them, we need to magnify them hundreds of times. There you have your average cell. Now, cells can go from anywhere from 1 to 100 nanometers. To put that in perspective, a nanometer is equivalent to 1/1,000,000th of a meter. Actually, let's put that into more perspective, a bit larger perspective. The diameter of the Earth is approximately 8,000 miles. Let's say that we made the human body the size of the Earth. A cell will be about the equivalent length of 2 feet. We can only observe cells with ultra-powerful microscopes, which magnify these cells hundreds of times. However, despite being really tiny, cells are also kind of big. Inside your average cell, you'll find a multitude of features like lysosomes, nuclei, ribosomes, endoplasmic reticulum, all is contained within that tiny cell. So, despite being so small, cells can hold so much. I guess you really can't say how big or small a cell is. It's all just a matter of perspective.
Hi, it's Mr. Anderson, and today I'm trying to answer that question that may puzzle you, and it's called, why are cells small? Why are all cells microscopic? And so before I get to that, I want to kind of start with a problem. So if we take a piece of paper, we'll call this paper 1, and we'll call this next one paper 2. And we were to roll them up into a cylinder. And so paper 1, I'm going to roll up into a cylinder like this. So we're going to call this one tall and skinny. And then paper 2, we're going to roll up like this. So we're going to go the other way, and we're going to roll it up like this. So we'll call that a little shorter and a little more round. And so the question is, which of those is going to have the greatest volume? So you may want to think about that for a second. This is a problem that I missed the first time I ever saw it. And so we've got 2 cylinders. I've kind of done it with the same, it's 8.5 by 11 paper. I've rolled this one into that tall and slender, and this one in the short and not so slender. And so the question is, which one of these has the greatest volume? So you might think 1 does, you might think 2 does, or you might, if you're really bright, might think that they're going to have the same exact volume. So there's really only one way to figure that out. We could do geometry, but what I'm going to do, since I teach biology, is I'm going to fill this one up with bark. A little bit more, top it off. Okay, so we'll say that 1, so cylinder 1 is filled up with this much bark. So now I'm going to take cylinder 2 and put that over the top, like that. And so if I pull out 1, a few things could happen. Number 1, if 1 has a greater volume, then it's going to overflow 2. If 2 has a greater volume, it's not going to quite fill it up. And if they have the same volume, it should kind of top it off. Or it might just fall apart and make a total mess. So let's see. Make sure you've locked in your guess. So I'm going to pull this up. So the right answer is that 2 has a greater volume than 1. So that might seem a little weird. I showed this one in class, and one kid accurately figured out that the surface area isn't the same because this one's going to have a greater surface area on the top than this one. But something happened when we went from something real tall and skinny to something fat and not so skinny. And you could imagine if we kept going and going and going, there'd be like a perfect object right here that would have the greatest amount of volume for the least amount of surface area, and that would be a sphere. And so there's a few things going on. One big thing is that we're trying to maximize the volume. And so in biology, if we ever try to maximize the volume, something's going to look a lot like a sphere. And so this is set to something called Allen's rule, and it applies to all endotherms, or all warm-blooded animals. And the idea is this: the farther you live from the equator, the more you're going to look like a sphere. And so if we look at early pictures, this is an early picture of an Eskimo family or an Inuit family, we're going to find that they're going to have shorter legs, shorter arms, stockier appearance. And the reason why, just like a polar bear, is that they want to maximize their volume because they don't want to lose a lot of energy in the form of heat. Likewise, when we go towards the equator, the people are going to look more tall and more skinny, and that's because they want to maximize their surface area. And so they want to get rid of heat. And so these are some Maasai warriors doing this jumping dance, and they're going to be really tall and slender. But it also gets to the idea of why cells are small. And so why are cells small? Well, by making them smaller and smaller and smaller and smaller, what we can do is we could maximize their surface area. So this is a bunch of bark, but it used to be one chunk of bark. And with that one chunk of bark, you had a relatively small surface area compared to the volume. But by chopping it up, now we have all the surface area on all sides of the bark. And so you know this, if you're trying to start a fire, it's important to cut that wood down to small bits, and so we can have reactions around the outside. It's much easier to get chemical reactions. It's much easier to start the fire. But likewise, our cells are small for the same exact reason. Our cells are small because we want to maximize the surface area on the outside. And the reason why is that we have to get nutrients into the cell, oxygen. We have to get waste products out, like carbon dioxide. And so if we can make our cells really, really small, then we have a large surface area. And also that oxygen then doesn't have to diffuse so far to get into the cell, or the carbon dioxide diffuse so far to get out. So that's why cells are small. If you're smart, you should be thinking, okay, if cells are small, that's good. Why aren't they infinitely small? Why don't we make them as small as they possibly could be? And the reason why is that if we make them too small, then we can't fit the machinery of the cell inside it. And so we can't fit the DNA, the proteins, and the machinery of the cell. And so the neat thing is that there's about a perfect sweet spot for all eukaryotic cells and a sweet spot for all prokaryotic cells. And they're all roughly the same size. And it all goes back to maximizing surface area compared to volume. And so that's cells, that's wood, that's cylinders, a little geometry, and I hope that's helpful.
Bacteria are the oldest living organisms on the earth. They've been here for billions of years, and what they are single-celled microscopic organisms. So they're one cell, and they have the special property that they only have one piece of DNA. So they have very few genes and genetic information to encode all of the traits that they carry out. And the way bacteria make a living is that they consume nutrients from the environment, they grow to twice their size, they cut themselves down in the middle, and one cell becomes two, and so so on and so on. So they just grow and divide and grow and divide. So kind of boring life, except that what I would argue is that you have an amazing interaction with these critters. I know you guys think of yourself as humans, and this is sort of how I think of you. And so this man is supposed to represent a generic human being, and all of the circles in that man are all of the cells that make up your body. So there's about 1 trillion human cells that make each one of us who we are and able to do all the things that we do. But you have 10 trillion bacterial cells in you or on you at any moment in your life. So 10 times more bacterial cells than human cells on a human being. And so of course it's the DNA that counts. So here's all the A, T, G's, and C's that make up your genetic code and give you all your charming characteristics. So you have about 30,000 genes. Well, it turns out you have 100 times more bacterial genes playing a role in you or on you all of your life. And so at the best, you're 10% human, more likely about 1% human, depending on which of these metrics you like. So I know you think of yourself as human beings, but I think of you as 90 or 99% bacterial.
I am going to show you the astonishing molecular machines that create the living fabric of your body. Now, molecules are really, really tiny. And by tiny, I mean really. They're smaller than the wavelength of light, so we have no way to directly observe them. But through science, we do have a fairly good idea of what's going on down at the molecular scale. So what we can do is actually tell you about the molecules, but we don't really have a direct way of showing you the molecules. One way around this is to draw pictures. And this idea is actually nothing new. Scientists have always created pictures as part of their thinking and discovery process. They draw pictures of what they're observing with their eyes through technology like telescopes and microscopes, and also what they're thinking about in their minds. I picked two well-known examples because they're very well known for expressing science through art. And I start with Galileo, who used the world's first telescope to look at the moon. And he transformed our understanding of the moon. The perception of the 17th century was the moon was a perfect heavenly sphere. But what Galileo saw was a rocky, barren world, which he expressed through his watercolor painting. Another scientist with very big ideas, the superstar of biology, is Charles Darwin. And with this famous entry in his notebook, he begins in the top left-hand corner with, "I think," and then sketches out the first tree of life, which is his perception of how all the species, all living things on Earth are connected through evolutionary history, the origin of species through natural selection and divergence from an ancestral population. Even as a scientist, I used to go to lectures by molecular biologists and find them completely incomprehensible with all the fancy technical language and jargon that they would use in describing their work, until I encountered the artworks of David Goodsell, who was a molecular biologist at the Scripps Institute. And his pictures are all, everything's accurate, it's all to scale. And his work illuminated for me what the molecular world inside us is like. So this is a transection through blood. In the top left-hand corner, you've got this yellow-green area. The yellow-green area is the fluids of blood, which is mostly water, but it's also antibodies, sugars, hormones, that kind of thing. And the red region is a slice into a red blood cell, and those red molecules are hemoglobin. They are actually red. That's what gives blood its color. And hemoglobin acts as a molecular sponge to soak up the oxygen in your lungs and then carry it to other parts of the body. I was very much inspired by this image many years ago, and I wondered whether we could use computer graphics to represent the molecular world. What would it look like? And that's how I really began. So let's begin. This is DNA in its classic double helix form, and it's from X-ray crystallography, so it's an accurate model of DNA. If we unwind the double helix and unzip the two strands, you see these things that look like teeth. Those are the letters of genetic code, the 25,000 genes you've got written in your DNA. This is what they typically talk about, the genetic code. This is what they're talking about. But I want to talk about a different aspect of DNA science, and that is the physical nature of DNA. And it's these two strands that run in opposite directions for reasons I can't go into right now, but they physically run in opposite directions, which creates a number of complications for your living cells, as you're about to see, most particularly when DNA is being copied. And so what I'm about to show you is an accurate representation of the actual DNA replication machine that's occurring right now inside your body, at least 2002 biology. So DNA is entering the production line from the left-hand side, and it hits this collection, this miniature biochemical machines that are pulling apart the DNA strand and making an exact copy. So DNA comes in, it hits this blue donut-shaped structure, and it's ripped apart into its two strands. One strand can be copied directly, and it can be seen spooling off down to the bottom there. But things aren't so simple for the other strand because it must be copied backwards. So it's thrown out repeatedly in these loops and copied one section at a time, creating two new DNA molecules. Now, you have billions of this machine right now whirring the right way inside you, copying your DNA with exquisite fidelity. It's an accurate representation, and it's pretty much at the correct speed for what it's occurring inside you. But I've left out error correction and a bunch of other things. This was work from a number of years ago. Thank you. This is work from a number of years ago, but what I want to show you next is updated science. It's updated technology. So again, we begin with DNA, and it's jiggling and wiggling there because of the surrounding supermolecules, which I've stripped away so you can see something. DNA is about 2 nanometers across, which is really quite tiny. But in each one of your cells, each strand of DNA is about 30 to 40 million nanometers long. So to keep the DNA organized, to regulate access to the genetic code, it's wrapped around these purple proteins. I've labeled them purple here. It's packaged up and bundled up. All of its field of view is a single strand of DNA. This huge package of DNA is called a chromosome, and we'll come back to chromosomes in a minute. We're pulling out, we're zooming out through a nuclear pore, which is sort of the gateway to this compartment that holds all the DNA called the nucleus. All of this field of view is about a semester's worth of biology, and I've got 7 minutes, so we're not going to be able to do that today. No, I'm being told no. This is the way a living cell looks down a light microscope, and it's been filmed under time-lapse, which is why you can see it moving. The nuclear envelope breaks down. These sausage-shaped things are the chromosomes, and we'll focus on them. They go through this very striking motion that is focused on these little red spots. When the cell feels it's ready to go, it rips apart the chromosome. One set of DNA goes to one side, the other side gets the other set of DNA, identical copies of DNA, and then the cell splits down the middle. And again, you have billions of cells undergoing this process right now inside of you. Now we're going to rewind and just focus on the chromosomes and look at its structure and describe it. So again, here we are at that equator moment. The chromosomes line up, and if we isolate just one chromosome, we're going to pull it out and have a look at its structure. So this is one of the biggest molecular structures that you have, at least as far as we've discovered so far, inside of us. So this is a single chromosome, and you have 2 strands of DNA in each chromosome. One is bundled up into one sausage, the other strand is bundled up into the other sausage. These things that look like whiskers that are sticking out from either side are the dynamic scaffolding of the cell. They're called microtubules, but the name's not so important. But what we're going to focus on is this red region. I've labeled it red here, and it's the interface between the dynamic scaffolding and the chromosomes. It is obviously central to the movement of the chromosomes, but we have no idea really, as to how it's achieving that movement. We've been studying this thing called the kinetochore for over 100 years with intense study, and we're still just beginning to discover what it's all about. It is made up of about 200 different types of proteins, thousands of proteins in total. It is a signal broadcasting system. It broadcasts through chemical signals, telling the rest of the cell when it's ready, when it feels that everything is aligned and ready to go for the separation of the chromosomes. It is able to couple onto the growing and shrinking microtubules. It's transiently, it's involved with the growing of the microtubules, and it's able to transiently couple onto them. It's also a tension sensing system. It's able to feel when the cell is ready, when the chromosome is correctly positioned. It's turning green here because it feels that everything is just right. And you'll see that there's one little last bit that's still remaining red. And it's walked away down the microtubules. That is the signal broadcasting system sending out the stop signal, and it's walked away. I mean, it's that mechanical. It's molecular clockwork. This is how you work at the molecular scale. So with a little bit of molecular eye candy, we've got kinesins, which are the orange ones. They're little molecular courier molecules walking one way. And here are the dynein. They're carrying that broadcasting system, and they've got their long legs so they can step around obstacles and so on. So again, this is all derived accurately from the science. The problem is we can't show it to you any other way. Exploring at the frontier of science, at the frontier of human understanding, is mind-blowing. discovering this stuff is certainly a pleasurable incentive to work in science. But most medical researchers, this is just discovering this stuff is simply steps along the path to the big goals, which are to eradicate disease, to eliminate the suffering and the misery that disease causes, and to lift people out of poverty. Thank you.
Tonight, I'm going to share with you my passion for science. I'm not talking about science that takes baby steps. I'm talking about science that takes enormous leaps. I'm talking Darwin. I'm talking Einstein. I'm talking revolutionary science that turns the world on its head. In a moment, I'm going to talk about two ideas that might do this. I say might because with revolutionary ideas, most are flat wrong. And even those that are right seldom have the impact that we want them to have. To explain why I picked two ideas in particular, I'm going to start with a mystery. 1847, Vienna, Austria. Ignaz Semmelweis was a somber, compulsively thorough doctor who ran 2 maternity clinics. They were identical, except for one thing. Women were dying of high fever soon after giving birth 3 times more often at one of the clinics than at the other. Trying to figure out what the difference was that caused this, Semmelweis looked at everything he could. Sanitation? No. Medical procedures? No. Airflow? No. The puzzle went unsolved until he happened to autopsy a doctor who died of an infected scalpel cut. The doctor's symptoms were identical to those of the mothers who were dying. How is that possible? How could a male doctor get the same thing as new mothers? Semmelweis reconstructed everything the doctor had done right before he got sick, and he discovered that he'd been autopsying a corpse. Had something gotten in his wound that killed him? With growing excitement, Semmelweis looked for any connection he could between dead bodies in the morgue and dead mothers in his delivery room, and he found it. It turned out that at the hospital with a high death rate, but not the others, doctors delivered babies immediately after autopsying corpses in the morgue. Aha! Corpses were contaminating the doctor's hand and killing his mothers. So he ordered the doctors to sterilize their hands, and the deaths stopped. Dr. Ignaz Semmelweis had discovered infectious disease. But the doctors of the day thought he was crazy because they knew, and had for hundreds of years, that odorous vapors called miasmas cause disease, not these hypothetical particles that you couldn't see. It took 20 years for Frenchman Louis Pasteur to prove that Semmelweis was right. Pasteur was an agricultural chemist who tried to figure out why milk and beer spoiled so often. He found the bacteria were the culprits. He also found that bacteria could kill people in exactly the same way that Semmelweis's patients were dying. We now look at what I want to talk about tonight in two ideas. We saw with Semmelweis that he was a revolutionary. He did it for two reasons. One, he opened our eyes to a completely new world. We'd known since the 1680s about bacteria. We just didn't know that bacteria killed people. And he also demolished fond ideas that people kept close to their heart. Miasmas didn't kill people, bacteria killed people. So this brings me to the two ideas I want to talk about tonight. One has opened our eyes to a completely new universe, and the other attacks long-held beliefs. Let's get started with Dr. Eric Betzig. He's a physicist who's opened our eyes to an entirely new world by violating the laws of physics. Betzig is a true rebel. He quit a job at a prestigious Bell Laboratory inventing new microscopes for biology because he thought scientists were taking his brilliant inventions and doing lousy work with them. So he became a house husband. But he never lost his passion for figuring out how to get microscopes to see finer and finer details than had ever been seen before or ever could be seen. This is crucial if we're ever going to understand how cells work and how cancer works and how something 150th the size of the head of a pin can do all these amazing things, like make proteins and move charges around and all of those things. There's just one problem. There's this thing called the law of physics. And part of the law of physics is a thing called the diffraction limit. The diffraction limit is kind of like when you go to a doctor's office, you can only see so far down, no matter how good of glasses you have. This was a so-called impossible problem. But one of Betzig's friends figured out how to take a tiny molecule that was smaller than the best microscope could see and get it to light up and fluoresce. Aha! Betzig said, I think maybe the laws of physics are not so breakable after all. So, he lashed together a microscope in his friend's living room. He had no laboratory. This revolutionary instrument got different protein molecules to light up in different colors. And with a computer, he was able to turn very, very fuzzy blurs into very sharp dots and produce images of unprecedented and startling clarity. For this work, last year, Eric Betzig won the Nobel Prize. Why? Because now we can see with unprecedented detail things that we never had seen before, and now doctors can get a better handle on things like cancer. But do you think Betzig was satisfied there? No. He wanted movies. The problem was that even the genius microscopes that he invented were just too slow. So what did he do? He came up with a 200-year-old idea called Moyet patterns. So the way that works is if you take two very, very fine patterns and you move them across each other, you will see a gross pattern that a microscope can see that otherwise you would not be able to see. So he applied this technique to taking a really blurry image of a cell and moving lots of structured light patterns across it until this cell became crystal clear. And here is the result. A mysterious new world, full of strange things zipping around, doing things that we don't know what they're doing. But when we figure it out, we'll have a better handle on life itself. For example, those green globs that you see, those things are called clathrons. They're molecules that protect other molecules as they move through a cell. Unfortunately, viruses sometimes hijack those to infect cells. Also, you see those little squiggly worm-like things moving around? Those are actin molecules. Unfortunately, viruses also climb down those things to get into the cell nucleus, to replicate themselves and make you sick. Now that we can look at movies of what's actually going on deep inside a cell, we have a much better chance of curing viral diseases like AIDS. So when you look at a movie like this, it's very clear that Betzig has opened our eyes to a completely new world. But he hasn't shattered any cherished beliefs. That leads us to Dr. Aubrey de Grey of Cambridge. De Grey definitely has scientists squirming with an interesting idea. We can be immortal. We can beat aging. Now, most scientists think he's a crackpot. Any Biology 101 student knows that aging is an inevitable consequence of living. For example, when we eat, we take in food and we metabolize it, and that throws off what we call free radicals. You might have heard of those, also known as oxygen ions. Those bind to our DNA, cause it to mutate, and cause us to get old and lose our hair. It's just like, no, it's exactly like oxygen binding to iron and making it rust. So you age because you rust out. Okay? Oh, and scientists also know there is something called immortality in cancer cells. So if you stop aging, all of you are going to turn into giant walking malignant tumors. These are cherished beliefs, but could de Grey be onto something? I think he deserves a closer look. First of all, I have a really hard time seeing him as a crackpot. Yeah, he started off life as a computer scientist, not a biologist, but he earned a PhD in biology from Cambridge, and he has published some very significant work on mitochondrial DNA and a bunch of other stuff. Secondly, he started an anti-aging foundation that has identified 7 different causes of aging, to me, that seem very plausible. And he is hot in pursuit of fixes for every single one of them. For example, one of the reasons we age is that our mitochondrial DNA mutates, and we get kind of old and our cells lose energy. He believes, and he's made a convincing case, that using viruses we can do gene therapy, fix that DNA, and rejuvenate our cells. One more thing. We have an existence proof that extreme longevity is possible. Bristlecone pine trees live 5,000 years. And some lobsters don't age at all. Now, this doesn't mean that the gray is going to revolutionize our lifespans. I mean, after all, we're not trees, and most of us are not lobsters. But I gotta believe that there are Darwins and Einsteins out there. And I'll tell you why. Consider this. There are 7 times more people alive today than during Darwin's time. There are 4 times as many people alive today as Einstein. When you consider that the proportion of scientists in the population has skyrocketed, there are now 7 million scientists. I gotta believe, and I do believe, that there's one of them out there who is working right now in obscurity to rock our lives. And I don't know about you, but I can't wait to be rocked. Thank you.